So we're ready to look at an overview of the eukaryotic cell cycle. So any cell that is actively dividing will go through these phases, and in each phase, which humans name these, so we broke it down into phases. For the cell, it's just a continuous process for them. But we break it down into these phases and we give them names, and then we describe what happens in that phase. So if you can list the phases in the correct chronological order, and you know what happens in each phase, then you know how the cell cycle happens. You know what everything you need to know about the cell cycle when it's occurring normally. So there are some variations on the cell cycle, but first we're going to learn the vanilla plain version. And then when you're in other courses, they may talk about some slightly different versions of the cell cycle that appear in certain particular organisms. But the general cell cycle has four main phases: G1, S, G2, and M. And cytokinesis is sometimes considered another phase, or it's the last part of, it's one of the subphases of M phase. So here it's listed, it's the last event. So I'm not going to get picky about that. But so G1 is called the first gap phase, but I like to think of G as growth, because this is where the cell is getting bigger. It's making macromolecules, it's making more organelles, so that when the cell does divide, each cell gets some of all of those structures. And so it has to build a whole lot of new macromolecules and structures. G1 is also the longest phase. Probably 40-45% of the whole cell cycle time is spent in G1. And if you look at a cell as it's in G1, under a microscope, you really can't see anything happening, not with a light microscope that we would use in lab, let's say. So it doesn't mean that there's not a lot going on in that cell. It's just, it's hard to see anything from our perspective. Then the cell will go into S phase. S stands for synthesis, but that's not helpful at all. It means DNA synthesis. So you just have to remember that. And this is where the DNA is replicated. And what we mean by that is every monad chromosome is replicated and becomes a dyad chromosome. So if you started off with 46 monad chromosomes, by the end of S phase, each of those monad chromosomes have replicated, so now they consist of two, but we don't call them two monads, we call them two sister chromatids because they're connected together, and so we call the whole structure a dyad. So 46 monads became 46 dyads. If you weighed the DNA, like on a scale, the DNA in the G1 cell weighs half as much as the DNA in the G2 cell, but if you count the chromosomes, the rule for counting is one monad chromosome is counted as one chromosome, but if it's dyad chromosomes, those are counted as one chromosome. And so you just have to live with that. But a G2 cell always has dyad chromosomes because G2 always comes after S phase. And what's happening in the cytoplasm is that the organelles are still, more organelles are being made, more macromolecules are being made. And so in terms of distinguishing between G1 and G2, just based on what's actually happening inside the cell, both of them have growth, both of them are making organelles and making macromolecules and the cell is getting bigger. What I would normally put kind of as a hint into like a test question, for example, is that in G1, the cell is preparing, not doing, but preparing for S phase. And in G2, the cell is preparing, not doing, but preparing for M phase. And so there will be some little hints in there to help you distinguish which one I'm going after, okay? If the cell is getting ready for replication of DNA or synthesis of DNA or replication of chromosomes, those are hints. But it's not doing the synthesis, it's just preparing. And the same thing with G2. If the cell is preparing for mitosis, preparing for karyokinesis, which is another name for mitosis. Let me write that down. Karyokinesis. Karyo kinesis. Karyokinesis means separation of the nucleus. Cytokinesis means separation of the cytoplasm. So you have a nuclear division, and then the rest of the cell divides. It's just the way it goes. So M phase, or mitosis, I mentioned before, some people call the whole thing mitosis, but mitosis, strictly speaking, is the fourth phase. It's the karyokinesis. And in a minute, we're going to subdivide M phase into 5, what I should have said, subphases. And these you've probably studied: prophase, prometaphase, metaphase, anaphase, telophase. And then the last event is cytokinesis, the separation of the cytoplasm and the plasma membrane, and if there's a cell wall, the cell wall, such that at the end you have 2 product cells. And those 2 product cells are genetically identical to each other, and they're genetic clones of the original cell that started the cell cycle. So by the end here, you have 2 cells exactly the same as the one cell that you started with. And so that's why it's a clonal cell division. It's just making more cells of the same kind. A lot of times, the cell cycle is artistically shown this way. It's a cycle, which means something you start with is also something you end with. In this case, it's a cell. You start with 1 cell, you end with 2, but those 2 can go back through the cell cycle and divide again. And so it's a cycle. And so they draw it like they draw any cycle, like a circle, because there's a circular nature to it. Now, the cell is not moving around. It's just sitting there doing all the events of G1 and then doing all the events of S and then doing all the events of G2. It's all happening inside the cell, at least at first. And then by M phase and cytokinesis, you can actually start to see a little bit of action. But it's always drawn in this circle, or what I sometimes think of as a clock or a pie chart or something like that. So in terms of a clock or a pie chart, G1 is the largest piece of the pie, or it's the longest section of the clock. It's the longest phase in actual time. It takes up about 40%, maybe 45% of the entire time of cell division. And so G1, a lot of things get done in G1. And then the next phase, S, is the second longest phase. That's where the chromosomes are replicated. And G2 is the third longest. So they're strangely in order by length of time. But that's just a coincidence. And then M phase is the shortest, if we include cytokinesis as part of M phase, which I typically do. I usually say I tag in cytokinesis as part of M phase at the very end, however you want to look at it. But in any case, the longest phase is G1. Remember, that's that first gap phase, but you want to think growth. S phase is where the monad chromosomes become dyad chromosomes. So everything in S phase is about the DNA, what's happening in the nucleus. Everything about G1 and G2 are what's happening in the cytoplasm. And then M phase is karyokinesis, division of the nucleus, followed by cytokinesis, division of the cytoplasm. And so we're going to see this diagram again, and you'll see similar depictions by other artists, but they always usually do this kind of pie chart or clock looking diagram. So if you haven't seen that before, you will see it in other places, other textbooks. All right. A chromosome in metaphase, actually, it doesn't have to just be in metaphase, a chromosome that consists of two sister chromatids contains proteins in between that hold the two chromatids together, and those proteins are called cohesin. All right. And then in this picture here, this is an actual micrograph where they stained the cohesin proteins green. So you can see them here lying between. What's showing up in blue are the two sister chromatids. So it's kind of interesting. And so the cohesin will hold those chromatids together until the exact moment when they are then released. So we haven't gotten there yet, but there's a cohesin between the two sister chromatids at the beginning of M phase, and that's where we're looking to at this point. So once the cell has grown in size and replicated its chromosomes and it's prepared for M phase, the cell then enters M phase. And M phase is divided into 5 subphases. So add the word sub in front of that. 5 subphases. So mitosis or M phase is a phase, and then we subdivide that one. And again, this is humans putting labels on events or periods of time in which events happen. And so once you learn the names of the subphases in the correct chronological order, and you know what happens in each phase, then you know the basics of mitosis. So we want to organize that, think about that as we're studying this. What are the names of the phases? What happens in each phase? And can we put those in the correct chronological order? So the subphases are named prophase, prometaphase, metaphase, anaphase, and telophase. And a lot of times I'll say telophase / cytokinesis. Like I mentioned before, I usually add cytokinesis as part of mitosis. That depends on your textbook, how they treat cytokinesis. But I'm not going to pick on that. Prophase. So what happens in prophase, I usually describe 3 main things that happen in prophase. Number 1, the chromosomes, which are dyads, condense. They get really tightly wound around those histones. And that's where you can really see them under a microscope clearly as separated structures. Then we have something called the spindle apparatus. The spindle is made up of these microtubules. You'll remember microtubules from when we discussed the cytoskeleton. And so during cell division, the microtubules have an additional special function. And a whole bunch of them will start to assemble, meaning be synthesized. And so the spindle apparatus or the spindle structure starts to become visually, you can actually see it under the microscope. So it's starting to be constructed. And then the third thing is that the nuclear envelope breaks down, or we say it disintegrates or disappears. And so those chromosomes are now no longer protected by that double membrane that we call the nuclear envelope. Those chromosomes are now exposed and accessible to the rest of the cell, which is really why they have to be really tightly wound around those histones, because that protects them. Okay, so that's the 3 events of prophase. If you missed it, rewind. Now, for prometaphase, which is the period between prophase and metaphase, this is where the spindle fibers are going to attach to a structure at the center of the centromere called the kinetochore. And so these fibers are attaching to the dyad chromosomes, and the way they're attaching, if you can look really closely here, here's one sister chromatid and here's the other one. And I'm going to try to draw this. This fiber is attaching to the sister chromatid that's on the left side, but this fiber is attaching to the sister chromatid that's on the right side. And the same thing is happening for this dyad, one fiber attaching to the sister chromatid on the left and one fiber attaching to the right. And so the fibers are attaching to individual chromatids such that one fiber is attached to the chromatid on the right side and one fiber is attached to the chromatid on the left. And those fibers are pushing and pulling on those dyads to get them to line up at the midline equator or metaphase plate of the cell. But in prometaphase, they haven't reached the midline yet. So the process of those microtubules attaching and then the dyads being pushed around is called prometaphase. When the dyads reach the midline, that moment when they were all at the midline, that's called metaphase. So metaphase is kind of this moment where all the chromosomes have reached the midline of the cell. And let me draw something here. So I will usually draw it, admittedly not very well, but I'll draw, there's a dyad chromosome, here's another one, maybe there's a little one. And what I will usually draw, let me get a different color. So this fiber goes to that one, to that one. Oops, it's hard to draw with a mouse. Okay, forgive me. That one, that one. I think you can see what I'm trying to do here. So when they're all lined up, you can see right here, the imaginary equator kind of runs right down the middle of all of these dyad chromosomes. But that's an imaginary line. You can see it, you can imagine it, it's that. But that's what the cell is doing. When the chromosomes reach metaphase, then they're aligned like that. And it's a brief moment, short period of time. Pretty quickly, the cell moves into the next event, which is called anaphase. In anaphase, the two sister chromatids for each of the dyads, and remember they're all in a line right now in metaphase, the fiber on the left starts to pull on the chromatid that's on the left, and the microtubule fiber that's on the right starts to pull. So they start to pull back. We say that the fibers, the microtubule fibers or the spindle fibers, retract or pull back. And what that does is it separates the two sisters from each other. And in order for that to happen, there has to be some removal of that cohesin, because remember that cohesin protein was that, in that previous slide, it was that green protein that lies between the two sister chromatids and holds them together. So there's actually an event that removes that cohesin so that when the fibers pull, those two sister chromatids can move away from each other. So we say that the centromeres are split. That's one phrase that we use. We say that the sister chromatids are moved away from each other. The dyads are split apart. There are several phrases we use to describe that. And you can see in this diagram here, all of these sister chromatids are moving to the left, and all of these sister chromatids are moving to the right. And yet they're not sister chromatids anymore. So as soon as the two sisters are pulled apart, we don't use that language anymore. Now you go back to talking about monad chromosomes. So you have this period in anaphase where a group of monad chromosomes is moving to the left, another group is moving to the right. And then telophase. This is going to be the end of karyokinesis. So in telophase, you really have the reversal of the three events of prophase. Remember, in prophase, the chromosomes condensed, they got compacted, more tighter. the nuclear envelope disappeared, and the spindle formed. Well, now the spindle is disappearing, disassembling. The nuclear envelopes are reforming, but now you're going to have two nuclei. So those nuclear envelopes are reforming, and the chromosomes inside of each nucleus are starting to relax. And then, for a brief moment, you have one cell with two nuclei, but then the cytoplasm is going to separate, and you'll end up with two completely separate cells. And that event is called cytokinesis. So cytokinesis has, in animals, it's simply a constriction, which is a tightening of a ring of actin filaments right under the membrane that tightens like a drawstring, and it pinches the one cell into two. So that's what they're showing here, is this tightening around the center here, and it keeps tightening and tightening until we have two cells. In plants, the cell has to build a new cell membrane in between and then a new cell wall because this is too rigid. The membrane can't pinch like this. So in plant cells, we have the formation of a cell plate. So let me show you that briefly. This is probably the most famous picture of a cleavage furrow, and you will see this exact same image everywhere. So I have to assume that nobody ever got an image this good ever again. But this is a kind of a famous picture of a cleavage furrow taken with an electron microscope and then artificially colorized orange. But that's a cell pinching off. And here's a cell on the right here that's almost separated. You can see it just has a little more to go. So two images of two different types of cells that are undergoing cytokinesis. These are animal cells. In plant cells, you will have, in the midline section, you'll have all these vesicles that bud off of the Golgi, and they'll line up and they'll all fuse together, and that will make the new cell membrane, and that's called the cell plate. And then later, the cell wall will deposit on the extracellular side of each of those membranes. So the cell plate is kind of like the early construction of a new cell membrane. And then later the cell wall is added. So we just went over a lot of the details for eukaryotic cell division. So if you are ready to move on, I encourage you to do so, or rewind and make sure your notes are complete.